Hi, everyone. I'm just waiting till everyone is joining. So I'll start slowly to the name of Katia, Boniface, and myself, Kurt Conrad, to welcome you to this month ESDR Kitchen. As you know, it is a program that has been started by the ESDR during COVID and maintained after it due to its success. We do now a monthly kitchen, which has different topics from freshly baked, which means recent publications, and then one of the more important ones also being the molecular cuisine, where really well-known leaders of investigative dermatology actually describe their discovery path. And this year we have the honor and the huge pleasure to have Eli Sprecher with us, who's going to talk about personal sequence. So Eli is not just the immediate past president of the ESDR, but during the time in the board and the exec, he has really become a mentor and a friend to his ability to take decisions and his kindness and everything. So it is really also a personal pleasure to introduce him here and give you a short bio sketch on Eli. So Eli Sprecher is the current CEO of the Tel Aviv Sourasky Medical Center, where he used to chair the division of the dermatology. That was not sufficient for him. So 3 weeks ago, he was elected and started as the CEO of the entire hospital. He's served also as the Deputy Director for Research and Development. He holds the Frederick Rice Professorship of Dermatology at the Faculty of Medicine in Tel Aviv, and as mentioned, is the past president of our society. He received his MD degree in Tel Aviv, in the Hebrew University, a PhD degree in molecular virology from the Hebrew University, and an MBA degree from Tel Aviv University. And he also did a postdoc in human genetics at the Thomas Jefferson University in Philadelphia in the United States. He has co-authored over 400 scientific publications and has several patents on some of these discoveries. He's going to talk about them in a few minutes. He has mentored, and I think that is also one of his key goals in life, to really nurture the new generation, and in that he has mentored over 100 and way over 100 students and residents, and his research focuses on genetic basis of skin diseases. His group aims at understanding the molecular genetics of both simple and complex traits, deciphering their pathogenesis, and then really translating them into this new clinic knowledge into the clinics. And that's, I think, is going to be the topic of his talk, of the personal sequence. So thanks a lot, Eli, for having accepted this invitation, and we're really looking forward to your talk.
Thank you very, very much. I'm just going to upload the presentation. Here we are. Well, hello everyone, and thank you very much, Cardine and Katia, for your kind invitation, and also for your very, very kind, maybe too kind introduction. As one of the founding fathers, actually, of the ESDR Kitchen, I must say that I feel particularly privileged to deliver today this molecular cuisine episode. Our life, of course, is a very long sequence of events, but I've selected some of those which may possibly be of interest to this audience. So I think that any sequence, of course, starts or must start with a very good story. And my personal story could have been a completely different story. I really wanted to, right from the start, as you can see here, to become a football star, like every Belgian child. But things turned, of course, differently, and it is always very difficult to say where everything started. But most probably, everything started in this old building in Brussels. This is the old Pasteur Institute of Brussels, where my mother used to take me during the weekends when she was on call. And that's where I saw for the first time incubators and petri dishes and even a huge electron microscope. I was indeed raised in Waterloo, Belgium. I moved to Israel in 1981, started my medical studies at the Hebrew University of Jerusalem, and I had the chance to meet there an extraordinary person, Yechiel Becker, who became my mentor during my PhD. And I was actually the third MD PhD in Israel. At that time, it was really not a common track in Israel. And I then served as an airborne physician in the IDF for a few years as part of my mandatory military service. And at the end of this period, I decided to become a dermatologist. Life and career, I think, are all about people, the people that you are meeting during your career path. And I was fortunate to meet two outstanding mentors, Ruben Bergman and Rachel Friedman, who really helped me find my way. And among other things, they sent me for a fellowship in human genetics. And this led to another unique encounter with Yoni Wito, who was my mentor during this fellowship, but remained also a very close friend until he recently, unfortunately, passed away. And also it is during this period that I started to understand the importance of colleagues who are also close friends. One of them is very well known to this audience, John McGrath, with whom I've been working or sometimes even running ahead all over those years. And then at the end, in 2008, I moved to the Tel Aviv Sourasky Medical Center, where I first served as chair of dermatology and then became VP for patient safety and research and development. I was also vice dean at the university, served as president of our society till last year, and 3 weeks ago, I became the CEO of my hospital. So this is in a nutshell my sequence. But what is perhaps not less important is an element which I think is critical to the career of any busy physician scientist. To me, it's actually the most critical of those elements, has been, still is, my family, to which I owe, I think, almost everything in my career, and to which I'm very, very deeply grateful. So apart from my family, my two greatest loves in life are skin and genetics. And with skin, I felt in love during my PhD studies, which were dedicated to the role of Langerhans cells in viral infections. On the left side of the slide, this is the first picture of Langerhans cells that I took in 1984. I kept this micrograph with me since then. My love for genetics was born out of my interest for rare diseases. As a young resident, I remember I was sitting in the clinic, and then I heard this patient complaining outside of my door of the waiting time. So I came to him, let him in. I was not supposed to see him, actually. Was scheduled for another physician. And this is where actually I was faced with my first case of genodermatosis. A patient was affected with a disease now known as Escherichia with papular lesion. And I discovered later on that the disease was caused by a mutation in the hairless gene, a finding which earned me my first significant paper in genetics. I was then fortunate enough to be awarded a very large grant from the Merck program, which encourages joint research between Palestinians and Israelis. And this was with a very wonderful collaborator, Moyen Canaan, and the rest was history. And since then, our group has deciphered the genetic cause of more than 20 genetic skin diseases and additional discoveries I will try to very quickly cover during the rest of my talk. Now, what has always fascinated me in genetics as a clinician has been the fact that genetics has given me the possibility to explain to my patients the cause of their disease. When a patient comes with hypertension and he asks you, what is the cause of my hypertension? I mean, there is really no good answers till today. But in genetics, things are very different. In some cases, you can actually become able to deconstruct impossible cases into easy to understand clinical presentations. And this is a nice example for this young boy of African origin, which was followed by Dr. Mubaya from Durban. And he was found to display a seemingly extraordinary clinical phenotype consisting of cognitive delay, and alopecia, and he has nail dystrophy and palmoplantar keratoderma and skin blistering. But this young boy was actually found through genetic analysis not to have an extraordinary clinical phenotype, but actually to carry 3 heterozygous mutations responsible for the unfortunate coexistence of 3 distinct, rare, but quite ordinary, well-known condition. He was carrying a BRAF mutation associated with a cardiofaciocutaneous syndrome, which could explain the alopecia and the cognitive defect in this young boy. He was found to carry a mutation in the keratin 66C gene, which is associated with pachyonychia congenita, and this, of course, is underlying the PPK and the nail dystrophy seen in the context of PC. And then he was found to carry a mutation in the keratin 14 gene, which is associated with EB simplex, which may explain the skin blistering phenotype. So this is a very complex disorder deconstructed into a very simpler clinical feature through genetics with obvious clinical implications. For example, in this case, the need for close cardiologic follow-up. Sometimes you have to deal with the reverse situation. This is a seemingly very simple case evolving into a surprising diagnosis through genetics. So this young adolescent was referred to as because of an ingrown toenail associated with suppurating parodontia. It's like perhaps the most common complaint that a patient can come to you with. I could ask the clinicians among you, among those clinicians who has not seen such a patient over the past month. But quite interestingly, the patient reported that a number of family members were also affected by severe and intractable paronychia. And upon physical examination, we found out that those individuals also displayed focal alopecia and trichomegaly and redundant plantar skin, as well as failure to thrive and facial dysmorphism and all kinds of neuropsychiatric manifestations. And then, of course, genome sequencing revealed here an heterozygous mutation in the HRAS gene, pointing to a diagnosis of a very rare subtype of Costello syndrome. But of course, more importantly, emphasizing the need for close monitoring for malignancies, and actually the father in this family was found later on to develop a bladder carcinoma. Now, from diagnosis, you can also move to treatment. One of the most common form of dominant hypotrichosis is hypotrichosis simplex, which is caused by mutations in the CDSN gene, which code for corneodesmosine. And very often, the causative mutations in hypotrichosis simplex are nonsense mutations. And then we set up an in vitro assay where fluorescence reflects actually the retroactivity due to aminoglycosides across the causative mutations. And using this assay, we show that gentamicin is able to promote read-through and to restore the biosynthesis of a full-length corneodesmosine protein in a dose-dependent manner. And based on this data, we treated a number of patients with hypotrichosis simplex with topical gentamicin with significant results. And this is really nicely illustrated, again, the power of basic research for a clinician who, through a good understanding of the underlying genetic basis of the disorder affecting his patient or her patient, can be key to the successful treatment of this disease. Now, another station along this sequence of events relates to what I learned from rare disease as far as common disorders are concerned. For example, the uncombable hair syndrome is exceedingly rare, as you may know. And in contrast, central centrifugal cicatricial alopecia, also known as the hot comb alopecia, is most probably the most common form of scarring alopecia among women of African origin. So this is very rare. This is very, very common. And the uncombable hair syndrome was found many years ago to be caused by mutations in a gene called PADI3. This is a gene which codes for the peptidyl arginine deaminase 3, which modify by citrullination very important components of the hair shaft, such as a trichohyalin in the iris and in the medulla, as well as S100A3, which is mostly expressed in the cuticle. And quite interestingly, we identified pathogenic mutations in more than 1/3 of CCCA patients, so now the common disorder, in 2 different cohorts, not only in PADI3, but actually also in the genes encoding its 2 substrates. So this is really connecting and learning from a very rare condition to a much more common one. Those PADI3 mutations were found to affect both the location, as seen on the left side of the slide, as well as the enzymatic activity of PADI3. And deamination was significantly decreased in cells transfected with a variant version of the S100A3 protein, is shown here by immunofluorescence. Now, quite interestingly, calcipotriol, a drug very well known to all of us, was shown to induce both the expression and the enzyme activity of PADI3, and this, of course, suggested the possibility to prevent CCCA in genetically susceptible individuals, and this formed the basis of a clinical trial run by 2 of our colleagues, Amy McMichael and Koza Dalova, in the US and in South Africa, respectively. Now, so far we have been speaking about rare diseases and complex disorders, but sometimes rare diseases can be quite complex as well. So EB simplex, I guess, is a disorder which is very well known to all of you. Is a typical example for a single gene disorder. Is very often due to mutation in the keratin 14 encoding gene, leading to blister formation within the basal cell layer of the epidermis. Now, EB simplex is a typical example of a single gene disorder, meaning that the disease is caused by one pathogenic variant in one gene. This variant being not only necessary, but actually sufficient to result in one defined disease phenotype. So this is the one gene, one phenotype paradigm. So you would then expect that, let's say, you have 2 siblings with EB simplex. They were born in the same family, exposed to the same environment. They are carrying the same genetic variant in the keratin 14 encoding gene. And those 2 siblings should display the same disease with the same degree of severity. But in contradiction with this definition, I think that everybody who has been working with patients with EBS know very well that very often you will see that a given disease phenotype like EB simplex can manifest with different degrees of severity among siblings carrying the same mutation. In some cases, you will have only involvement of the plantar surface, for example, and in other cases, much more widespread involvement. So we assembled a series of EB simplex families with siblings displaying different or divergent degrees of severity. And we found out that the more severely affected individuals were found to carry pathogenic variants in the gene called HMCN1. Now, HMCN1 codes for a protein called emicentin. And we found out that emicentin is expressed at the basal cell layer. And in addition, we found out that emicentin emicentin-1 also directly interacts with keratin 14. Now, given the importance of keratin 14 for the pathogenesis of EB simplex and the co-segregation of emicentin-1 variant with a more severe phenotype in patients with EB simplex, and even the fact that emicentin-1 interacts with keratin 14, we hypothesize that emicentin-1 may perhaps be essential to the stability of the epidermal-dermal junction. And in order to ascertain this possibility, we generated 3-dimensional organotypic grafts. So these are small pieces of skin that you can grow in a 3-dimensional fashion in Petri dish. And you can take punch biopsies from those artificial pieces of skin. And then we compare those 3-dimensional organotypic grafts with grafts grown in HMCN1 deficient, in emicentin-1 deficient grafts. And we've consistently observed the formation of subepidermal blisters, which nicely replicate the human phenotype. And then it actually turned out that emicentin-1 actually serves as a scaffold for the formation of the keratin 14 cytoskeleton, not only demonstrating a major role for emicentin-1 in epidermal-dermal junction function, but I think more importantly suggesting that screening for emicentin-1 variant may be of relevance to the analysis of families at risk for EB simplex. So this is, in a nutshell, a short summary of a very long sequence. Of course, I could not walk all that path alone. I would like to very much thank all the people who have been part of the various projects from my lab that I mentioned during this talk, my many other colleagues at the lab and outside the lab, or funding agencies, and of course, all of you for your very kind attention. Thank you.
Thank you very much, Eli, for this really nice talk and for sharing your journey with us today. So to remind people, you can ask your question in the Q&A. So maybe with Cuisine, we will start while you write your questions. So I had a question. So I'm not working on like rare disease, but I was wondering in the field of dermatology, whether like gene therapy with like the CRISPR-Cas, for example, is it a perspective for patients with this kind of disease that you showed, or?
Of course. I mean, I think that many genetic therapies are of great relevance to all of those diseases. The thing is that there has been a lot of attention and work and efforts and resources invested in developing those tools, which are working remarkably well in vitro. The major problem with the skin is, of course, the delivery issue. And so then you have the, always the choice between systemic deliveries, which can be associated with unwanted side effects, and the challenge of getting transdermal delivery, which is, of course, a much bigger issue. There have been some attempts which have been successful, but still, I think that we are far from a situation where we could actually have a platform that can universally be used to deliver those technologically very sophisticated solutions. So I think that at this moment, another path which is of great importance, as has been actually it has been very well demonstrated in many contexts, is repurposing of existing drugs. And so, for example, the short example that I gave regarding the use of topical gentamicin to treat patients with hypotrichosis simplex is a nice example. There are many similar examples, such as, for example, the use of statins to treat patients with Pachyonychia congenita, and additional examples like those. So there are, I think, two major strategies which should be looked at.
Okay. After I had another question. So regarding what you were showing on the EBS and said like one mutation, one phenotype, but actually it's more complex than that. What do we know about the influence of the gender? Because right now we are talking a lot about in research taking into account like also the gender, like what can it make as a difference? And do you observe in the clinic, or when you look at the like gene from some different like polymorphism, for example, between like women and men, or like depending on the age, also the hormone status, or?
Yeah, I think that there are actually very good examples showing that there are some diseases which are genetically more penetrant in one gender as compared to the other. For example, CCCA is a disease which is much more common among women than among men of African origin. It's still so you can explain the ethnic propensity based on the fact that those genetic variants for example, in PADI3 are much more common in the African population as compared to European population, but it doesn't explain the gender propensity. It can also, of course, be associated with some exogenous factors. Some people have been discussing the role of traumatic injury to the hair follicle as a triggering factor, but there are a lot of discussion and controversy about that, even though the disease is sometimes still called outcome alopecia, but it's most probably a misnomer because in some cases it's very difficult to associate the local injury with the development of the disease. So certainly there is, of course, much more to be looked at in terms of gender differences. I agree.
Maybe just hooking up on that quickly about penetration and diseases that actually get along life's journey either are attenuated or even getting worse. Do you think that epigenetics, did you look into that epigenetics plays a role, that some diseases actually quiet down or others just getting more severe?
Yeah, certainly. So, you know, there are a lot of epidemiological observation. I just heard this morning a lecture about PTSD, and there are even experiments in rodents showing that 2 generations after exposure to a trauma, so the grandchildren of those traumatized animals are still showing a behavior which is consistent with PTSD. So there is certainly some epigenetic factor which are involved. We have, for example, a nice published a few years ago, a nice example where we found in a certain gene just one mutation. It was a gene mutation in the calpain 12 encoding gene. And the second mutation could not be found at the DNA level, but could be found at the RNA level, was a 40 base pair deletion. And the reason for that was actually hypermethylation of cryptic splice site, which was activated and led to this 40 base pair deletion, and actually had been inherited from the mother of this patient. So there is certainly, again, a lot of things to be looked at the epigenetic level. It's always, I think, a question of availability and cost and also expertise with the technologies. But I'm sure that once this becomes much more readily available, and also the bioinformatics behind it become more widely available, there will be a lot of those so-called unknown mutation which will be uncovered.
And then maybe a bit more personal approach question, because you were talking about repurposing of drugs and approaches. How do you approach problems in genetics? You said you brought the disease of the uncombable hair and then the centrifugal hair loss. Do you look for maybe diseases that look similar, that have similar traits, and then you go into the genetics? Or do you say like, well, I'm just searching in fishing in the dark, and I might dig up something which then resembles something else? How do you approach your ideas? Because they're really broad.
You mean in the context of drug repurposing for more specific?
No, that's just, repurposing, but also in general, if you say like, I want to look into a new project or a new disease.
I see.
You like your first patient, you said like, well, he got to have a genetic defect.
Actually, it's mostly, so first of all, we have to suspect, of course, that something is genetic, which is, of course, always a question that you can answer, you know, looking at, so if there is some inherited trait, so that must have some genetic component. And also there are some clinically relevant questions. So if sometimes you have a patient or a group of patients who are extremely active, and they will drive you into research. And I think that a wonderful example for that, and this is a topic which I didn't touch, but has been also a very important component of my professional path, has been my contact with patient support group, and one more particularly the Archeonica congenita project, which is an amazing endeavor, which was started many years ago by Mary Schwartz, and she was helped by Francis Smith and Irwin McLean at that time, and they really drove a lot of scientists into that project, which actually is extremely active, and I'm confident that they're acting like a kind of a catalyst, and they are driving all very outstanding scientists into trying to find a solution for those patients. And this interaction, this interface between patients and scientists and clinicians is something which is unique and outstandingly powerful. And so that's something which I think also is sometimes leading your choices.
Thank you.
Okay. Maybe I will ask the last question, so which is quite personal, because you mentioned at the beginning that you perform your PhD on Hungarian cells. And so after you moved to like genetics, completely different, or at some point, did you think of trying to link the two? Because I'm sure like with all the genetic disease, you have a lot of influence of the immune system also that has some deregulation. So do you miss like Langerhans cells, even if it's been a while now, or are you trying to link the two?
Of course, I miss them. But beyond that, we have a large, actually connecting immunology and genetics. So we have a large program on pemphigus vulgaris, which, as you may know, is a disease which is particularly prevalent among Jewish populations. So that's why we have access to many patients here. And actually, our first focus was on trying to look at the skin and trying to look within the skin what is actually predisposing a patient to develop pemphigus vulgaris. But more recently, we have switched and actually identified something which looked like a major player in the pathogenesis of pemphigus. And the paper is just currently under writing, but it's so we are actually connecting, I think, the skin and the immune system from a kind of a broader perspective. I think that autoimmune diseases or autoinflammatory diseases, the attention has been mostly focused on the immune system, looking at the epidermis as a kind of a more inert, passive victim of the immunological attack. But there is a lot also to be looked at the response of the target organ. In our case, it's of course the epidermis of the skin. And that's what we are trying to do. So connecting the skin and the immune system in the context of pemphigus. So I have a connection.
Okay, great. Thank you again, Eli, for this really nice talk and for sharing your personal sequence. So as a reminder for everyone, and you can tell the people who were not able to attend that webinar will be broadcast in a few days through the YouTube channel of the ESDR. And so next slide, please, Leah. So now just to finish, so this was the last kitchen webinar for 2024. And so we will have a new series of kitchen webinars that will start in January with a freshly baked. But also we will have, so starting next year, so a fifth flavor, so the ASID flavors, which will combine so both the ESDR, the GSID, and the SID. So we thought it would be nice to have a broader audience also with all the American and Asian people, and so almost like people from everywhere in the world, of course, who are invited to see. And so our next ESDR Kitchen webinar will be with Ruti Lubin and Raquel Salcaro. So on dendritic cell subsets in health and inflammation, and also on Langerhans cells and autophagy. So two really recently published journals. So thank you all for being here today. And so we will see you again next year. And just to finish, yes, we will have in September from the 10th to the 13th, so the 44th annual ESDR meeting, which will be in Antwerp, Belgium. So you will have soon more information on the ESDR website, but you can already book the date on your calendar so we can see all each other in person in Antwerp. Thank you. Thanks again, Eli, and happy holidays, even if we have some more days before. Joyeux Noël, as we say in French. Goodbye.
Bye.